Hello everyone, welcome back to our systems biology podcast. Today's podcast interview session reminds me one of my school days event. One of our teacher was teaching and he told us that every one of us is a magic. He was teaching, actually, he was drawing on the blackboard the embryonic development from how a single cell, all of us originates, fertilized cell, and that single cell become two cells, then four cells, then a lump of mass of cell, like a ball. And then all of a sudden, just like a magic, different organs develop, and eventually you and me get birth. It's pure magic. And in fact, one few of the earliest stalwart in biology who thought about systems level understanding of biology were actually developmental biologists. And developmental biologists have always contributed majorly in understanding biological theories, phenomena, and they have done beautiful experiments. By that, we right now understand lots of genetics, cell biology, molecular biology of development. But still, certain things are elusive. Still, developmental biology, embryonic development is an enigma. Is there any law, universal law, that governs that development across different species? Is there any design principle that is implemented in those molecules that we know today? These questions are still elusive. Starting from the day of the famous developmental biologist Weddington, we are still trying to develop underlying theory or physics behind how we got birth, how we got this whole body. So discuss with this interesting enigmatic topic, we have with us Dr. Archishman Raju. Archishman is a faculty at the National Center for Biological Sciences, NCBS Bangalore. So welcome Archishman to our podcast.
Thanks. Yeah, thanks for inviting
Thank
me.
you for taking out time on a weekend to spare some time with us and with our audience. So, without further ado, let us jumpstart with some questions, right? So, as I understand, you work on developmental biology related problem, and as I was saying initially, that it is still an enigma. Every day when I see myself on the mirror, I find it enigmatic that how it could happen is the magic. So, as a scientist, as a scientist working in this interdisciplinary area, tell us one of the magical questions that still haunts you, that drives you in this field. And why do you find that question is so problematic to explain and answer?
Yeah, thanks, Biplab. And in fact, you're right that, well, not only were some of the early developmental biologists, I guess, pioneers of self organization and pioneers of thinking about complex self organized systems in biology, but if you go back even earlier, in biology, people often in history of biology talk about mechanism versus vitalism, where the vitalists were people who thought that there's a vital force and that you cannot explain the explain biological things somehow physically. And these people were in fact developmental biologists because they were equally the vitalists. Many of the famous vitalists were developmental biologists because they basically said, how could you possibly have a physical mechanism for what you're describing as magic? So in a certain sense, magic is the right word for even looking at the history of developmental biology. Well, I mean, I guess if you look at fully, and I should clarify, I guess, that I'm trained as a physicist, so I'm not trained as a developmental biologist. But so my interest is primarily in thinking about quantitative approaches to understanding developmental biology. And so the questions that animate me may not necessarily be the questions that animate a person who's doing experiments in developmental biology. But I think that well, there are several remarkable things, as you're saying, about a single cell turning into a fully formed organism. I think one of the mysteries about it, if you like, that still um, amazes me is the robustness with which it happens. So, you start off with a single cell, then the cell divides, and then eventually, right, cells specialize to take very different functions. So, for
Every
example,
place, the right cell goes in the right place in the right time.
right, right. So, if you think, right, a neuron is very different from a skin cell. but your brain doesn't suddenly have a little bit of skin cells in there, or right, your heart doesn't have a little bit of lung cells in it. So this organization is very specific and happens very robustly. And I think the how different cells end up taking up different fates, why a neuron becomes a neuron versus a blood cell becomes a blood cell. I think this we understand quite well now, in the sense that it has been studied extensively using techniques from molecular biology, and we have a pretty good idea of why different kinds of cells look very different and are very, or at least take very different fates. But I think what's more harder to understand is how the whole thing comes together. um, and you in the end get this very complex self-organized organism that if you have five fingers, you have four fingers and a thumb, you get five. You don't, actually sometimes you can get six, but very rarely.
Most
uh,
of the cases not.
yeah,
Overwhelmingly, most of the cases not.
yeah, yeah. And if you think about it, it's a very, very hard problem to make sure you always get five. But it's similarly, right? Your head is not too big. So these things happen very robustly. And as I was saying, the early, one of the things that really um, was interesting to the very early people who were doing experiments was that you can take, say, an embryo when there are just four cells, so just at the four cell stage, after two divisions, and you can separate these cells, for example, in a sea urchin embryo, and then each one of these cells is capable of becoming a fully formed organism. And how that happens, how that happens, I think we still don't really deeply understand. Obviously, we have a much better understanding of all the parts that go into it, but I think we still don't really deeply understand the robustness of the system to changing the genetics of it, changing the environment in which it happens, but even generally, um, even in normal conditions, how does it manage to get everything right? Yeah,
If I just try to expand what you are saying, because
yeah,
many times students get confused with that, because we say that we know lot about the molecular and cell biology of developmental biology, but
yeah.
still something is missing. So
Hmm.
possibly it is like that, that we know lots of implementation of certain rules. We
Hmm.
see that implementation, we know lots of implementations, that how the different molecules are used, but what is the generic rule behind it, that the
Hmm.
cell is trying to implement, so that we
Hmm.
get it robustly every time the process work. So overall
Hmm.
rules of the game we do not know, but we know
Hmm.
lots of parts of it which are involved. We can manipulate those parts and play with it, but we do not know as a whole the what is the rule of the game that is holding
Hmm.
things together, right? Can I say that way?
Yeah, that's. I think that's a fair description, and there are also. In recent times, people have started worrying about not just the parts, but also things like the geometry, things like the mechanics. But also, these are not necessarily rules in the sense that they're also, or if you like, they're stochastic, right? So these are noisy processes. So how all of that comes together, yeah, is we don't fully understand. Um,
Okay.
how all of
Yeah,
that
yeah.
comes together robustly, we don't fully understand. Yeah,
Okay. So if I continue with this, so you must be working on multiple angles with this problem. So certainly must be this must be open problem. So if you can tell our audience, who which are mostly undergraduate, postgraduate students of different areas. So if you can explain what type of open problems you and your group is working on right now.
hmm yeah. So one of the things that we have been working on is precisely as you mentioned. C.H. Waddington was an early developmental biologist who was quite formative to the field, and in many ways um, he was prior to the all of these things that we're talking about, right? The idea of molecular biology, meaning we discovered really what kind of proteins are responsible for different functions that cell, right? Different differentiation of cells into different types. What kinds of proteins do that? What kind of genes do that? We discovered this pretty late. This was the late 1970s, 1980s, and going forward. Now we know a lot, but Waddington was working in the 1930s, 40s, 50s.
So
up
in
until
fact,
the
when he was
70s.
working and doing experiment, we were not aware about the DNA makes the gene and all these things.
Right, right. I mean, so DNA
So he
itself
was
was
post
discovered.
genome time,
Right, 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 right. So, yeah. So he was
pre genome time,
yeah,
pre
or pre-molecular time, definitely. I
pre
mean,
molecular time.
yeah, genes were known in the Mendelian sense, but yeah, we didn't know the structure of DNA, and we
Yeah, we
didn't.
were not
Yeah,
aware
right.
that what that thing
Yeah,
is.
yeah, exactly. So
That's
because
actually
of
very
this,
amazing. See, you do not know anything about the molecular level. You can
yeah,
manipulate, you can see things,
yeah, yeah,
and you are doing abstraction, isn't it? Just pure abstractions you are doing, mind
yeah,
bending abstractions.
yeah,
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 please
and
continue.
no, and it forced people to, I think a little bit theoretically about the whole problem, because the research project was not to figure out all the parts, because you really didn't have a way to figure out all the parts. So you had to try and figure out what was happening, as you're saying, abstract it away somehow from the parts. What Waddington, one of his fundamental ideas was that what I was describing earlier, these cells ending up taking different fates, right, becoming different types of cells in the body. He visualized that idea in terms of an abstract landscape, and the landscape he imagined having valleys and ridges, and the decision of a cell he thought of as a ball rolling down this landscape, and in the end there were various different stable points in the landscape where the ball could eventually
So it
come and
will
land.
fall from a higher point to a lower point.
Yeah,
That's what you are
yeah,
saying.
yes, yes. And in the end, wherever it lands, so there will only be a few pockets for it to land. So there'll be valleys and ridges in between, and those will correspond to different types of cells. And actually, Waddington himself wasn't even just thinking of this landscape for cells. He even thought of it in terms of organs, or even he thought that organisms themselves could have different kinds of phenotypes which were discretely defined. So I mean, it was an abstraction. So people were obviously worried. What is this landscape? What does it represent? He had a debate actually with Crick, of Watson and Crick fame about it. Crick was not very convinced by it, and for a long time it was sort of, the ideas didn't really get much traction in the field, because the field moved in the end towards, a molecular biology understanding. But I think more recently, and particularly with the advent of stem cells, which people have now gone back to this picture, but it's still being used just as a picture. And so my work has really been part of an effort to try and convert this picture into mathematical description, to to say that, can you mathematically write down these landscapes in
I
a
mean,
way that
you
can.
want to write down some sort of function or equations to represent this landscape, which still biologists are using just as a metaphor.
That's right. Yeah. Yeah. Exactly. And then can they describe actual decision processes that are taking place during development quantitatively? Because usually, right, you think of these processes qualitatively, in the sense that one cell becomes can become two differentiate into two different types of cells. But can they really describe them quantitatively? What's the path that a cell takes to go to, to differentiate into other kinds of cells? So that's been one focus of my research. I mean, I'm also a little bit, actually, more recently I've gotten into, just to give another example of something I'm interested in, I've gotten interested in how different decisions are timed, right? When does a cell decide that it's going to become a different type of cell? So obviously we know that embryonic development takes some time, and one process that's happening is cell division, right? You're just getting more and more cells. But then the other process that's happening is this specialization of the cells into different types of cells. And so you can imagine the various different things that are timing this process. And there are two kinds of timers you could imagine, right? You could imagine a clock, something that oscillates, or you could imagine something like an hourglass, right? This which sand glass timer, right? Which then falls down, and that's another way of trying to keep time. And turns out both of these processes
Okay,
are
you mean
very.
in one classical our wrist watch, grandma's watch, it is the oscillatory thing which
Yes,
is actually maintaining the time, giving me the rhythm, whereas in
yes,
this case, yeah, that's interesting. I never thought of that.
yeah,
Yeah, it's a
right,
completely
right.
different mechanism.
Yeah, so
Yeah.
these are two different mechanisms, and turns out that both of them operate in development, and how they
Oh, both
operate,
of them operate?
yeah, both, and sometimes both of them can even work together.
Uh-huh.
So anyway, so these are interesting and actually lesser studied questions, because spatial pattern formation people have thought about a lot, but temporally how do patterns change is has been less studied. So that's another
And
thing that
in fact, if I remember a little bit of biology, this
yeah.
temporal pattern is also extremely robust.
Mm.
Rarely things goes wrong, right? Rarely, it's like spatial robustness. Temporal
Yeah.
events are also very precise in the right time it happens actually.
Yeah, yeah. No, so in fact, one good example of it for several organisms, for example, for small worms, if you change the temperature, it's well known that they will still develop robustly, but the time that they will take to develop will change. So,
Okay.
if you increase the temperature, it will shorten the time. If you reduce the temperature, it'll take longer time. And in fact, recent research is showing us that what you're saying, temporally this happens in a robust way. So if one event happened at let's just say, just to make it up, right, one event happened at 5 seconds, second event happened at 10 seconds at one temperature, then at another temperature, if the first event happens at 10 seconds, the second one will happen at 20 seconds.
Uh,
So you get
so
this kind of,
scale is same. Scale
yeah,
invariance is there.
yeah, yeah. So these are very interesting questions, how this is achieved, because if you imagine temperature is something that changes the chemistry of everything,
Yeah.
and there are all these things happening, there are all sorts of, right, and so you would imagine that everything changes, but then how do you get something that happens so nicely? So these are the kinds of questions I'm interested in.
That's very interesting. That's very
Yeah.
fascinating. So
Mm.
essentially, in your lab, you primarily work on approach discussion from mathematical and physical perspective, right?
Yeah.
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 Yeah. So as I understand, as you also mentioned just a few minutes back, you are actually a trained physicist. And if
Yes.
I remember, you had your PhD from Cornell, and then you moved to Rockefeller for your postdoc stint for a short while, and then you moved to India to NCBS again. So now, as I was looking into your old paper, so essentially the broad area of your training during PhD was primarily statistical physics, if I say broadly, right?
Hmm.
Renormalization
Yes.
and another thing. So, and then you moved into this classical problem of biology, one of the hardest problem in
Mm.
biology. Actually, everybody has to say that, right?
Mm.
So, what made you decide that I'll move into that? What is that or that moment? Was there any particular moment where you decided, or something happened in your life that you decided, no, I want to change the gear and jump started completely different? What you even physicist term, you are changing
Mm.
the regime. You are almost like phase transition. You are going from one regime, one state to another state. Actually, is there any such thing?
Um, well, I guess I was always interested in biology, mainly really out of my, reading, I guess, popular science books, particularly on theories of evolution. And there used to be several. I mean, biology actually is one thing that there are several popular science books about. Often they're well written, um, and but I think the field that perhaps gets the most coverage in popular science, I mean, I don't know for sure, but at least the one that I read the most was on evolution, and that always was a very interesting idea for me, to, right, just think about the diversity of form that's arisen out of these quite fundamentally simple ideas of evolution. Um, and um, uh, but of course, the kind of coursework I saw in biology, to be very honest, was not attractive to me. Just like for many students of physics, it's not attractive.
Yeah, that's
And
true.
yeah,
Even
so
the school level teaching is also that way, is many a time is not attractive.
yes,
That's a
yes.
limitation we have actually.
Yes. So then that's why I did my undergraduate in physics. And when I did my PhD, I was already looking for biological problems. But I had did my joint PhD in a physics department. And one of the ideas I got exposed to um, in the group I was in. So as you said, I was working on really statistical physics, but even statistical physics in many ways has broadened and been now applied to many different fields. Right? It's not the traditional statistical physics of gas in a box. It's really
Yeah,
become quite a
from
broader
finance
field.
to biology everywhere.
Yeah, yeah, and so. One of the problems that the research group that I joined had been previously working on was actually to look at models in systems biology. And what they had found was that so many of these models in systems biology have many components, right? And the dynamics of these components is described by differential equations, and this is quite typical. And there are many parameters,
For
and
which
what
you do
they
not know the value.
of which, yeah, which are usually unmeasured. And so what they had found was that actually there is a sense in which most of these parameters don't matter, which is why they started calling these sloppy models.
Yeah.
um, and so the idea was that there are if you think in terms of the individual parameters, then it seems like all of them matter, but actually you somehow have to rotate in the space of parameters. You have to think in some rotated way, and then there are some directions that matter more and other directions that matter less. Now, these might
Right.
not be the parameters that you chose originally, but there might be some combinations of them, some complex
Ah.
combination of them. So that idea was quite interesting to me, and I guess it attracted me in the sense that you had this right complex biological system which you could try and understand in ways that were more interesting to a physicist or somebody who comes from that perspective. Um,
Yeah.
yeah,
Yeah. So this brings me to one interesting thing is actually when this is a as a systems biology teacher, and as I interact with both biology, physicists, everybody, and give talks in multiple disparate forums, right? This is a struggle because see, when I talk to my friends in physics department,
yeah,
for them it is very they are very happy to accept that idea that okay, your model has just two parameters or something,
yeah, yeah,
right? Simple
yeah,
model, they are very happy. But when
yeah,
I show a simple such a parameter, less parameter small model to a
yeah,
biologist friend,
yeah,
they always struggle with that, this idea that how could you make it so simple. See,
yeah,
like these there are hundreds other molecules, there are so many interactions are there, why are you disregarding them, discarding them, it cannot be, right? So this
right,
is, I
right.
think, it a struggle every one of us face to in all our discussions. So but from your training in physics, you have learned to believe and appreciate that even a very complex system need not to have a very simple solution.
Hmm.
Sorry, a complex solution. A
Complex
simple
solution.
solution could be
Yeah,
can be there, right? So this
yeah.
idea that as I was looking into your web page also, so you were saying that, as you were saying here also, that you want to focus on simple models,
Hmm.
right, for complex systems like developmental biology, right?
Yeah, yeah.
So two questions comes to my mind because many a time students get confused with this, particularly people who are coming from biology background. What the hell do you mean by model in this case,
Hmm. Hmm.
right? And why do you believe that a simple model can actually, if we make it correctly, can actually capture such a complex phenomenon? So can
Yeah.
you just explain it for our students?
Yeah, maybe I'll take a little bit of time with this
Yeah,
question, and just start by motivating, because there are many ways to answer it, but I just want to start by motivating it by right the again from personally the I got it right as you were saying. My PhD was in this idea of the renormalization group, and that's not something that's even many physics students are not necessarily taught about. So, but just to at a very coarse level describe it, and then I'll come back to this model question and why it's important to it. um, I mean, the renormalization group is a very so already in physics there was right you already had complex phenomena happening in materials. So for example, the phenomena of superconductivity, even though you understood electrons in metal, or people thought they understood the behavior of electrons in metal, somehow superconductivity was not it was not easy to understand superconductivity. And so in physics already there was a sense that when you get complex systems, right, and this is in non-living materials, uh, the way you describe it, it was not clear how do you describe it, because the traditional approach in physics was always a kind of reductionist approach, that you try and right reduce the system to its simple to its parts and then figure out the behavior. But I think superconductivity was an example where this approach didn't seem to work, and eventually people realized that you want an what is this idea of emergence, the idea that actually the theory of the phenomena of many component system can be an emergent theory, and often that theory can be independent of the details of the system, the parts of the system. And so the theory of superconductivity doesn't depend on which particular material is a superconductor, right? It's formulated in ways that's somehow, I mean, obviously it depends on certain properties of the materials, but it's not similar to the theory of how we understand rigidity nowadays. The fact that solids are rigid objects doesn't depend on which material is making the solid, but is a more kind of general theory about how this rigidity is also a pretty surprising phenomena, right? You push a table and all the molecules in the table, they all maintain their distance and then move in the same way rather than getting disorganized. So so in physics, this tradition had, I think, come in the what is called condensed matter physics, that you have to explain complex interacting systems are explained with theories that often don't depend on the parts that constitute the system. Uh,
Microscopic detail of the whole thing.
right, and the microscopic details of the system. And then the renormalization group was an idea that really formalized this, and I won't go into the details of it, but it really told us in a deep way that there are some parts of this, there's some if there's some things that matter and some things that don't um, matter as much. And more interestingly, um, the so okay, so that's one thing. Then I'll come back to this question of model versus what does it mean to make models.
Simple.
Yeah, and simple models. So one idea is that you just make a simple model because it's a choice. So model is, you can think of as an idealized abstraction, right? That of the phenomena, which ignores many details of the phenomena. That's at a very general level. And one idea is you choose to ignore details of the phenomena as the choice of the modeler. And that's a legitimate idea, and that's one way to think about it. But I think the renormalization group in physics at least gave a much, a kind of deeper understanding of this, where it really said that there are some parts of the phenomena. If you're interested in explaining some things, you don't have to. There's some things you can ignore, and some things you have to pay attention to. And that's what made it such a powerful idea. So, to give you a simple example, I mean, to give your listeners a simple example, right? The idea of diffusion, as being described by the diffusion equation, is not something that depends on whether it's ink in water or perfume in air. And but if you really think about it at the level of the molecules, these are very different
All
processes.
different.
Yeah, but the fact that at somehow large length and time scales you can describe them all by one equation, the diffusion equation, uh, is in a sense an example of that. Is an example of how you can make theories of phenomena that are simple, even though the underlying constants may be complicated. And now in physics, we sort of have a way of doing this. In biology, we don't really. But this is, in a sense, the challenge. And this is why many people are interested in trying to approach biology from this perspective. Can we figure out somehow in a precise way what kinds of models we need to make for complex biological phenomena? Or is it that somehow the models have to be complicated? You have to make a big model with all the parts, and that's the only way somehow to think about the system. So that's the sense in which I think people, right, want to are interested in making simple models, because it's not just that you're choosing to ignore details, it's that you hope that if you think about certain collective phenomena of the system, then it won't depend on the details in some precise way, and you will be able to figure out what it really depends on. And that's a challenge always, and that's not something that's very clear how to do across biological systems.
So
Um,
if I
yeah,
transgress a bit from here, see,
yeah,
as I gather from you that, uh, as you said, this is a challenge that, okay, how can I look into the details, biological details, and
yeah,
then distill it,
yeah,
and then actually abstract a simple idea.
yeah, yeah,
Right. So if I understand, rather than just having mathematical acumen here, the people who works on this type of problem, they have to be very good in this abstraction thing,
hmm,
right? That you have to do some abstraction. And I don't know what will be your opinion on this. I always find this interesting thing. See, if you look into the
yes,
early biology, like even if you look into Mendel's work, right? He does not have any idea about gene or anything,
yes,
right?
yes,
But
yes,
he is doing abstraction that there is something which is discrete,
hmm,
and that discrete thing gets shared. Essentially, the binomial equation is getting.
yeah,
So the whole thing is an imagination.
right,
He has no evidence that there is any discrete component that is getting shared, right?
yes, yes,
And what I find that if you look into early biology historically, if you see, we
yes,
were doing lots of abstraction, studying evolutionary biology and everywhere. Even in 30s, 40s biochemistry literature, there are so much of abstractions actually,
yes,
right? And
yes,
one interesting thing is connected with physics also. See, in physics, you can suppose you think about astronomy, right? How the universe got created and all these things. You can observe the stars,
yes,
but you cannot manipulate to do the experiment to understand it, right?
yeah,
So, isn't it like that? Earlier biology was also like that. I can observe, but I
yes,
do not know how to manipulate. So, I was doing abstraction. I was bringing mathematics to that. But
yeah,
then one day molecular biology came. I can manipulate it,
yes,
and all of a sudden we lost the tradition.
yeah,
What do you feel
yeah.
about it?
No,
Do you
I
think
think
there's
that's
something
no,
historically that
that's
is happening?
yes, definitely. And in fact, in developmental biology, right, what we just discussed, this Waddington's landscape is an idea, is an abstraction. And in fact, Waddington was very aware that the underlying parts have to be complicated because at that point you didn't have molecular biology, but you still had genetics in the sense that you could make crosses and right. Uh, you had sort of early genetics was already there, so he knew many genes could be involved in making this landscape. But see, he still abstracted this idea from the phenomena, from observing that when you make crosses or when you uh, that you can't do. But even today, now when we can do genetic manipulations, you still seem to get the same types of cells. You still don't rarely get right half a skin cell and half a neuron. That's not something that you see. So it's it was very much part, I think, of early biological thinking. I agree with you. Which the advent of molecular biology definitely. In fact, theoretical biology as a field almost doesn't exist anymore. Right, a biologist
Yeah,
today is almost by definition an experimental biologist. But at
yeah.
that time, there were people who considered themselves theoretical. And in fact, Waddington was known for organizing these theoretical biology conferences uh, in the early 70s, which were trying to, yeah. So yeah, I agree with you.
But do
Yeah,
you think now a new trend is coming up because the it seems with time things are changing, because as I see many of my friends and colleagues who are from engineering background, physics background are picking up
right.
problems in biology,
Yeah,
and they see that has happened earlier also. Many people from other background moved into biology, but many of them eventually got dissolved into this the field of cell and molecular biology, experimental biology. But
right,
still, right now, I see lots of my friends and colleagues. I see, in fact, sometime back, last 2 years back, I think I did a meet online conference where almost 14 people were there who essentially work on, abstract models and all this type of thing, right? So, do
right.
you think a new trend is coming up where biologists are also appreciating that we should not look just only on experimental thing where we will manipulate a cell or a organism, generate new data, from that data I'll go into another experiment, but as a whole, people have started appreciating, at least one section has started appreciating, and that is expanding, that we have to build abstract theories which can be predictive models, which can
Hmm.
be written in mathematical sense. And then just like physics, the experiments will be driven by those models, not
Hmm.
the opposite one.
Right.
So do you think that type of trend in the academia at least happening?
I mean, to some extent, I mean, definitely it's probably more than before, but I think there is still a lot to be done. Uh, and
Okay.
in many ways, I find that even some of these early conversations that were happening um, between mathematicians, physicists, and biology, for example, at these theoretical biology conferences that I just mentioned, were more free flowing, more sort of deeply engaging with each other. And I mean, obviously, now you have many more physicists who are in biology, and so there is much more work that is being done, definitely. But I still think that there is scope for more genuine engagement between biologists and physicists and mathematicians. And so, in that
So
sense, it's an open place where I think we can do more.
So, what do you think is the major bottleneck? It is because how we are training our students, or somewhere else?
I think that's part of it. I think partly it's also there can be quite different disciplinary cultures between biologists and physicists, and sometimes it's on both sides to try and bridge those cultures, um, and I think. Yeah, so I think partly it is that not just in terms of training students, but also in terms of the way these fields have become defined, particularly in the past, let's say, 60 or so years, has contributed to that. But I think definitely, I think there is a lot of possibility of change, and so in that sense, I think it's an exciting place to be in because you can actually, right, rather than being in a field where everything is fixed and already, right, there is one path. It's a field where you get to decide and get to partly contribute to the direction of um, where things will go.
Yeah, that's encouraging, because see, many a time what I see that, as you said, there are, in terms of popular science books, a
Yeah,
lot of easily available popular science books usually deals with biology related problem, medical science related problem. So young minds usually get excited with that. Although
hmm,
eventually, because of multiple other reason, I see
yeah,
many of our students land up in hardcore engineering or physics or somewhere,
yeah,
but they have enjoyed always, although they may not have enjoyed learning biology in the school level, but
yeah,
these questions, right? So I see many of these students join our elective courses, they interact with us, right? So I hope many of them will be seeing these videos also, right? So
mhm,
if I say, ask you, can you tell, apart from this developmental biology thing,
yeah,
can you tell us some of the open problems which are very challenging, where
mhm,
actually we have to put some young mind to do these abstractions, and bring mathematical and physical concept, physics driven concept to that. So if you
right,
can pinpoint some of the, open, maybe must be there a lot of open problem, some of the
yeah.
other open problem, if you can highlight.
Yeah. Well, I think so. Just a little before I do that, one of the reasons I think that well, I know particularly physics students tend not to want to go to biology is because right, physics is somehow seen as this field that does fundamental science work, and then biology is seen as, right, something that's more somehow derivative or more right, somehow lesser of a field. And it's interesting what we are discussing about. It just occurred to me, what about models? Because in physics, often you're taught laws, right? Especially at the undergraduate level. So Newton's law of motion, second law of thermodynamics, law of conservation of energy. And it's interestingly, once you get to the PhD level, then it all shifts to models, even in physics. So if you're in cosmology, you'll do the lambda CDM model. If you're in particle physics, you'll do the standard model. If you're in
Yeah,
condensed matter
so in
physics, you'll do
science
the
everything is a model actually.
so you'll do the Ising model, right? And I don't know if anybody today will can name anything really that they would strictly define as a law.
Right.
in the sense that it was understood. And so these are these older philosophical assumptions about what constitutes science that are still with us. And I think in our pedagogy, we've not done enough to, uh, you know,
Break
bring
these
in
barriers.
these ways that science has changed. And we've realized that actually, right, the second law of thermodynamics is not really fundamental. It comes from course training, statistical mechanics. Newton's laws fail, right, of course, in many regimes. Even conservation of energy is a consequence of symmetry. It's not a fundamental. So all of these things now are seen in very different ways. And somehow I think in our pedagogy we have to show that
Yeah, in fact,
you know
I'll interrupt. I think
yeah,
we do not also teach students that all these models are actually provisional,
yeah, hmm,
right?
yeah,
And
yeah,
every day we are trying to break and make new models.
yeah, yeah,
That's what we are doing.
yeah,
Somehow
yeah,
we do not communicate that to our students. Actually,
yeah, right,
as if these are fulcrum of truth, and we have to
right,
use
right.
them. That's how we teach them.
Right, because we've inherited somehow philosophical right presumptions of the 17th century Europe, or whatever, that we are still continuing, that have gone become very outdated. But I think and the thing in physics that we understand really is, which really makes physics different, is we know what model, if you like, applies in what regime, and we sort of know in many cases how to connect and how to understand at some length scale, right? But can you imagine if we needed to know quantum electrodynamics to figure out how hydrodynamics, right? It would be a hopeless hopeless scientific task. But then somehow we have a sense that at different length scales we can do this, at other length scales, other regimes, we can do this, and so on. This is the right theory. In biology, right, of course, we don't quite have something like this. And it brings me one of the challenges in biology, that it's not clear that things separate out so nicely, right? So, for example, something like what everybody is familiar with, right? This COVID virus that happened, right? It was something that was a mutation. It was quite something at the smallest scales that affected the larger scale, right? The literally
Ah, that's
society.
beautiful. Yeah.
Yeah, right. So it was,
Somewhere one single base mutation has changed
yeah,
the whole history.
yeah, change everything. Yeah. And so it's not clear that one can study the large scale necessarily without paying attention to the heterogeneity at the small scale. And in a certain
Yeah.
sense, that is reflective of the challenge of dealing with biological systems, where we and why, in a sense, they're harder. And so, I mean, in terms of specific problems, honestly, the problem is there are too many rather than that there are not enough. And more of a challenge is picking the right problem, right? Origin of life, how the brain works, developmental biology. So there are problems in all of these fields in terms of figuring out quantitative mathematical description. But I think, broadly speaking, from a physicist perspective, what is exciting in them is that biological systems are, of course, out of equilibrium, right? So these are non-equilibrium systems, and these are evolved systems, right? Which is why they have a very specific kind of heterogeneity, which is not random. They're not homogeneous, but they're also not quite random. And finally, right, there may be other ideas, like many people now think about information theory, the ideas of information and how information gets transmitted, that might be specific to understanding biology. So this is at a very broad level, but I think really figuring out, in a sense, how non-equilibrium evolved systems work is a very very difficult challenge, and something many people are engaging in from various different directions.
Okay. So before I go into this next question, see,
Yeah.
as you said this, do you feel that uh, where the biological system as a complex systems exist as we see it, that maybe traditional tools of mathematics and physics is not really helping us to get a grip of it? Because,
Mhm.
for example, the example that you gave actually mind blowing. I'd say one single mutation changed the history, and it may have happened so many times in our evolutionary history on Earth, right?
Yeah. Yeah.
So
Yeah.
one single mutation is actually at a single mutation level, it is a quantum event possible,
Mhm. Mhm.
right? and that's a completely different regime the mutation is happening, whereas the changes that is happening as large time scale.
Yeah,
And
yeah,
as you said, in physics we learn to separate out this time scale
yeah,
completely. You have different law for the other one time scale,
yeah,
you have different law, and we don't know how to connect them. We do
yeah,
not try to even connect them.
yeah,
We say
yeah,
we don't try them, right?
mm,
So that actually gave me this thought immediately right now, that maybe we require a different type of physics approach or different mathematical approach
mhm,
to actually
mhm,
get hold of this problem.
mhm,
That there is heterogeneity, there is uncertainty, but at the same time it is robust,
right,
right? And
right,
it is evolving. It is not a static one. So,
right.
do you think we need different tools, conceptual and mathematical tools?
I think so. I think so. I think there are some people who approach biology from the perspective of, we have tools from physics, so sort of we have the hammer. Let's find the nail, and that's, I think, the wrong approach. Really, what is really is that we have this phenomena, which is so complex and interesting, and can we figure out the right mathematics and physics to describe it? And it hasn't, I mean, it's an open question. I think it requires a lot of sustained effort to then that again, that's why there's an exciting field.
Yeah, that's actually good, because then
Yeah,
we need more young minds to actually start early and chase this type of thing, rather
yeah,
than one particular biology problem, possibly some
yeah,
generic approaches to capture this, right?
yeah,
So
yeah.
this brings me to my last question in this discussion. See, you did a graduation in physics, then following any other physics student who are good in physics, moved to do PhD, and then eventually you, because of your interest, you moved into this intersectional area where you're trying to work on development biology. Now, as I said, many students I encounter who are interested,
Hmm.
right? That they want to explore new areas. They do not want to get stuck in their classical engineering or physics or math background. So,
Hmm.
from your own perspective, own journey,
Hmm.
can you give us some, I can say, suggestions or advices to these students, that how should they, okay, if they have some rudimentary interest and they have a dream that they want to make a big in this,
Hmm.
right?
Hmm.
Moving out of their traditional career opportunities.
Hmm.
So, how should they prepare themselves?
Yeah. I think this is multifaceted question. I mean, at a practical level, what helped me was doing a summer internship, and I did a summer internship when I was in my undergraduate years, where I actually just did something very random. It was in fact at NCBS, and I was studying the. I just picked up some book and I read through the chapters, a book on evolutionary dynamics, and I was influenced by this chapter. I mean, I got interested in this chapter on the evolution of language, and so I got very interested in that question and trying to model it and so on. And then eventually, by now, of course, in all these years, I think it was. I was not, I don't like to do that kind of modeling anymore. But at that time, it was formative. It was right.
Yeah,
It was. Uh, so it was. So I think, and getting that opportunity in a summer internship was definitely very helpful. But I think there is also a broader kind of systemic question to this in what people see as a traditional scientific career. And I think, even if you think about it, these ideas of rigid disciplines is a very, very modern idea. You really don't see it before the 20th century. And even really in the second half of the 20th century is really where it gets solidified, is clear departments with their own bureaucracy into, and if you
that's
take
right.
some of, yeah, if you take some of our own modern scientists like a JC Bose, he was not defined by, disciplinary
No
boundaries.
way. No.
Yeah,
Yeah.
and I mean, many, you take any of them, Mahal Nobis, Meghnad Saha, Didi Kusam. I mean, there are very wide range of interests and
They
uh,
essentially chase the problems,
yeah, yeah, and they were
not a
interested in
discipline.
knowledge, right? They were interested in
Yeah.
knowledge. They were not interested in, right, a particular. But unfortunately, the structure of academia today has become one where these people would not survive, honestly, because they would not be publishing enough papers to get into there. In a sense, I think it can be hard in today's academic system. And then, so of course, then we are going back to interdisciplinarity after first creating disciplinary boundaries, where actually, if you think about it, prior to that, it was already a much more wider approach that people were taking. And so Even though I think it can be sometimes hard in academia because it has become geared towards right producing trained technicians who are specialized, um, and they're very good at writing papers in their specialized fields. I think there is a lot to be learned from this previous generation of scientists and their very broad idea of what doing science meant. And I do think that that's something to keep in mind and try and pursue as far as possible, because I do think that's where knowledge has to go. This is becoming a little unsustainable, where you just become so narrow and so specialized that in the end of the day, uh, you know you're not being creative anymore. You're not allowing yourself full expression of what drove you there in the first place, right? In fact, I see a lot of students who come and right. I was just having this conversation with students who said that, yeah, when I come in my first year, I want to cure cancer, and right by the fifth year, I want to publish in some journal. Right? And so you can see the right. And I think the first one is a much more worthwhile. Of course, you can laugh at it and say it's naive and so on, but it's a much more worthwhile spirit to have, to pursue something for knowledge rather than to pursue something for this very specific.
So I think you'll agree that to
Yeah,
some extent, although the realities of the world is hard, but
yeah,
if you have to do good thing in science, it is
yeah.
better to be childish and naive to some extent, and follow the dream adamantly
Right.
rather than losing it, isn't it?
Yeah, I mean, yeah, to have that spirit. Yeah, I agree. To have that spirit of discovery, to have that, and I think naturally, then you start appreciating questions that are not so rigidly confined, because that's where often I think, especially today, in many ways, so many of these disciplines have become so saturated and so advanced that I think the creative space often is in thinking broadly. Yeah, I think definitely having that spirit of youth, or I guess if you like, is something a lot of people dismiss idealism, but I don't. I think it's good to have idealistic people. Uh,
Yeah.
yeah, yeah,
Yeah, in fact, that's how you can actually survive better in this hard real world. If you
yeah.
have some, something to bank on, a dreamy idealism and all this. Yeah,
I
that's a
agree.
nice question, nice observation. Because
Mm.
students has to get grade, they have to pass the exam, but at the same time, if you lose our what excites us most,
Yes,
right, then it is difficult, even, right. So
yes, yes. And I,
I
I
think.
should say, sorry, I should say that that's also partly on us, that we should have spaces that can nurture that idealism,
Yes,
rather than
yes,
always
that's true.
try and suppress it with
Rigid
whatever
boundaries and
rigid
targets and
boundaries.
all these things.
Yeah, yeah,
Yeah, that's interesting. That's interesting. I think
yes,
our audience will take this in the right spirit, right? So that they can actually do not lose the dreams they started their higher education with,
yeah,
and try to grab the best opportunity to chase that dream,
yeah,
right? To wait
yeah.
for the right moment and prepare themselves for that.
Mm,
Okay, Chisman, thanks a lot for spending the weekend with me here and
mm,
discussing these diverse topics. These are diverse and interconnected, and sometimes very difficult to communicate in just one half an hour discussion or something. But I firmly believe many of our students will get encouraged by your journey, looking into the questions that you are working on and the other questions that you said, and the last one that you said, that you should not, neither the teacher neither the student should kill the dream of and the spirit that we have, right. So
mm.
I will end this discussion with that spirit, that the eternal child that we have, the child that always asks question in the school, sitting
Mhm.
and asking the question to the teacher, will survive among all of us. Thanks a lot for joining, and best of luck for your research.
Thanks, Siddharth.